So is this all those 6 papers on structure-activity relationships, and I kind of perceive them as an extension of the previous material, because so far in all those papers from the 60s and in the general retracement of this road that was sketched out in the first of all in the lab books, and in addition to that in this series of scientific chemical papers, there were like minor, let's say, generalizations, and usually it was a description of particular chemical compounds discovered and analyzed with properties to some extent, but there were no generalizations on this mm, let's say structure activity relationships uh, from a distance, comparatively speaking, yeah, how various general modifications result in what type of activities. And this was first done, to my knowledge, in '69 for the first time, yeah, in 2 papers. And then he started developing this SAR analysis, and all those subsequent papers are more and more insightful, rich in material and in observations, in various insights, structural insights, or structural activity insights, which is something that did not occur earlier in the 60s. So I think the SAR analysis was first started in '69. Yeah, and then so we have like a decade of papers, and one of them is no, 2 of them are published in '69, 2 of them in 1970. Then a few years later, there is this '78 paper. All of them have this structure activity relationship in the title. And finally, in 1994, there is one more written with Peyton Jacob III. And this one is short, but so full of insights when you compare, and so rich in material, because Shulgin is after having examined all those tryptamines. And even in the family of phenethylamines, there is much more discovered, right? So suddenly the amount of material is way larger than before. So and the suggestions, the SAR analysis are also kind of developed even further. So there is an interesting progression of both thought, theoretical thought, and empirical material, right? There's progress in both. So those papers are kind of progressively deeper and richer. And so this is interesting to compare them. This is one thing. So I think we should first have a look at the first one, see what's there, and then see the progression and perhaps compare it to some extent. And then another thing, well, is that they differ also in style, in the way they formulate it, the way they classify, but those are superficial differences, right? And I think this is very interesting according to my knowledge, this was never then repeated by Shulgin, and even in Tikal and Pikal, such general analysis are never undertaken, right? There's only an essayistic part, the introductory first part, and then the second part is only kind of empirical. I would say it's a description of each particular compound, alphabetically listed, but never kind of grasped in a structural relationship between other compounds from the family of either phenethylamines or tryptamines. Yes, this dynamic structural relation that exists between each subtype of a family of compounds is never repeated in those books like The Nature of Drugs, or which is a series of lectures, or Pical and Tikal. So those articles that we are discussing right now, I think, are of utmost theoretical importance. This is the highest kind of theoretical insight that you can have in the relation between all those various substances that were synthesized or discovered by Shulgin. Would you agree? Or so this is a kind of higher layer of theory superimposed upon this merely empirical description of a given substance and its activity. What do you think?
I think they both have value in different ways, and I think it was intended that Pical and Tikal are not so theoretical, so comprehensive, but rather show the essence, the nature of the experience, and how it integrates into the lives. Whereas, indeed, as Sasha mentions or hints at many moments, his drive or why he did the chemistry the way that he did, very systematically exploring how the change in the molecule results in a change of effects on the human mind, and then putting it all together into some kind of a map.
Yeah.
Although he did mention himself that it's not complete at all, and that others will simply will have to continue because it's so vast. But this gives like a preliminary first foundation for such pursuit. So I believe, in a way, yes, they're different, and this is perhaps in regards to really focusing on the comparison or correlation of how the structures and their changes are comparably or can be compared between one another, or see some developments of, if I extend this chain here, this is what seems to happen, or a trend develops here. And if we add higher molecular weights atoms on this position, this is a trend that seems to happen, and so on. I agree that those papers, those organizing reviews that we collected for the seminar, are perhaps the most informative on that aspect. But I would not say that either is higher or more valuable than the other. It's just simply taking a look at this comprehensive and very multi-layered field from different angles.
Yeah,
and also, I read this very randomly, this piece in Tikal, I think is the appendices at the end of the book, where he himself admits that there's just so many angles to look at the area of psychedelics, and he is taking the chemicals point of view and perhaps going substance by substance, and from there branching to the biological effects, the effects on human mind, the culture or anthropological aspects, the biology where these substances occur in nature, if they do. But he admits there is all the other possibilities, and one can go from biology and say this plant is used in this way and contains these chemicals, and this is its history, and so on. And it's so beautifully written, and it's certainly true. This kind of overview will never be complete, and there is never going to be someone to put it all together. And this is just one point of view that we can take.
Yeah, you're right. You're right, but there are various perspectives. Not only chemistry is the only one we agree, but even from the perspective of chemistry, I agree also that he takes various perspectives, but it's not about one of them being kind of higher in regard to value, but I would say that's what I suggested is theoretically more kind of interesting or more theoretical because of this distance that kind of grasps the entirety of the field of the it has a higher scope of looking at all the substances from a various type or class at once and sees the relations between them, not just one kind of substance or but a certain class or so the attempt is a kind of total complete synoptic view at the entirety of the field and to see the relations between the particular types of compounds, right? So this is what I consider theory, at least in the Greek sense. Theory is kind of looking uh, at a certain discipline or domain of knowledge, yeah, or domain of reality, and looking at it and describing it in its entirety. So we have like two kinds of perspectives. One is from the distance, this kind of theoretical distance, and another is close up. You describe a particular compound. Yeah, are you there? I think you're saying something, but not to me, apparently. Yeah.
Oh, yeah, no, I'm here.
So yeah. So this kind of theoretical distance is of value here. I think a very valuable analysis, and it has to be done on the basis of empirical material that you gather first, right? So I think in the 60s he first in the 60s, starting in 1960 starting from the first synthesis that he did, for 1 decade he gathered empirical material which was described in the lab books and in the publications from the 60s. And then 69 and 70 is the beginning of this theoretical analysis from this theoretical distance, where this empirical material collected, mainly phenethylamines, right? Tryptamines were done later. We see in the first articles from 69 and 70 that he had not yet a lot of experience with tryptamines, and he has not synthesized them so far. It was his second attempt that he did after the phenethylamine. So and he admitted that in one of the articles, I think he has this. I think let me see. It's an in the 1970 article on the slides, you can see it in Chemistry and Structure Activity Relations of Psychotomimetics, Article 60 in Shulgin's enumeration. Yeah, he has this division of simple monocyclic biochemically plausible compounds that could be synthesized by a human organism, that could be a part of human metabolism, and then which is mescaline analogs, that is phenethylamines, right? And this is what he starts with because it's simple to analyze, right? Because of the simplicity of the structure. And on the oh yeah, there's Mark. And on the other hand, he says there are complex polycyclic, metabolically alien compounds, right? So So this is interesting that he starts first with those that are not metabolically alien, which is why he starts with phenethylamines. First, because of this simplicity of this structure, which makes them prone to analysis, and on the other hand, because of this fact of them being metabolically kind of or biochemically plausible and not alien like the other compounds, the ergotamines, the tryptamines, and so on. So this explains this order.
Vitamins also are designed so they're neurotransmitters in the brain just as well. But also another consideration, I think, is because phenethylamines are somewhat easier to work with chemically.
Yeah.
And that could have also influenced why starting there.
Yeah, because of being simpler structurally, right?
Yes, the chemical transformations are straightforward for many of phenethylamines, whereas not so straightforward for tryptamines, although it just depends case by case.
Yeah. Agreed. So shall we continue in the way that we envisioned, paper by paper? What do you think? What do you think?
Also, hello, Mark. Thanks for
Hi.
joining now.
Hi, Mark.
Apparently,
are you
Good
there?
morning. Sorry, it took me a while.
Yeah. So we have prepared a presentation with the material. Did you have a look at it already? I think the material in this document on Google Docs.
It's
more or less
an impressive syllabus.
Yeah, it's more or less the same material. So it's like a summary of the 6 papers on structure activity relations, right? So the idea is to have a look at those 6 papers one by one and discuss them as a kind of development of Shulgin's theoretical glance at the relations between all those compounds that he encountered, right? Because there is a clear progression of theoretical insights that he had. Would you agree?
Yeah, definitely.
All right.
Same.
So let's start. The first paper, where there are 2 papers from '69. We could start with either the one that is most straightforward, which is the psychotomimetic agents related to catecholamines, or the other one, structure-activity relationship of one-ring psychotomimetics, which is kind of dense, I would say, and perhaps let's do them one by one, and then proceed to other papers. So what is here? In this, maybe let's start with psychotomimetic agents related to catecholamines. Okay. So first, catechol, a benzene ring with 2 hydroxyl groups, and a catecholamine, which is the same one with an amine group on the right. Yeah. So examples of catecholamines: dopamine, noradrenaline, adrenaline, and so on. And so he gives a division into 3 classes, out of which I think he focuses mostly on the phenethylamines here, right? In most of those papers, but here in this first one, he also mentions tryptamines and compounds which include the various atropines. So it's the nightshades family, right, which he dislikes and distrusts. So he does not, and they are of course also complicated structurally, so not easy to analyze. Therefore, he rather does not focus on them. Yeah, and so acetylcholine related agents like atropine, scopolamine, and others, they cause confusion, delirium, amnesia, and so on. So they are not psychedelics. So they are mentioned here, but not really analyzed. What is analyzed is tryptamines and phenylethylamines, which should be here. I think here this should be phenylethylamines. This title is misleading, and of course,
I think you had it. It looks like he's just kind of laying out classes of drugs that make you trip.
yeah, generally.
Two of them are psychedelic. One is a true, the anticholinergics, sometimes called true hallucinogens.
Yeah, yeah, right.
But I think you have that title right. Like those are the serotonin indoles, and he's just kind of seems to be laying it out.
Yeah, so he mentions them generally, but I think in those early years of his research in the 60s and 70s, he does not really focus on tryptamines, and in those he mostly focuses on phenylethylamines. And so then, oh yeah, so the title is fine. You're right. So he says that psilocin is already active, DMT is not, unless you have an monoamine oxidase inhibitor, which we now know perfectly well, and this is what he established. And so these are just basic claims that you can have on serotonin type agents. And then he proceeds to the other substances like phenethylamines in this case. But what he knows, it is interesting to see how few tryptamines he knows at this point, right? He knows psilocin, he knows DMT, he knows 5-MeO DMT, and this is it. Yeah. So in comparison with what he describes in those later papers from, for example, in the 1994 paper, there is, after having written Tikal, there is such a big difference that here we can see he did not really have much experience with tryptamines at this point in 69 or 70, right?
Or he's just, it's not the focus of the paper, and so he's just kind of, like DMT and blah blah blah.
Yeah, not the focus of the paper, but if you look at the lab books, it was not his focus at all at that time, I think. So not yet, not yet, and which is fine, which is, I think, kind of assumption or a program that he had. He had to first examine the phenethylamines and then tryptamines. So this was his conscious decision to do it in this order. I think this is how it looks like. And so
it's just growing out of kind of his early mescaline experience.
Yeah,
and that structure catches his eye, and it's
yes,
of the make the most substitutions the most easily by far over the tryptamines. I mean, I guess forgetting about the
for the moment, yeah. And so he has certain principles, structure activity principles, and what are they? They are introduced here and in this paper. So side chain length. I think the first analysis that he did was to analyze those TMA family of phenethylamines, which are constructed out of mescaline, right? And comparing them, he developed various general conclusions, let's say, structural characteristics, structure activity characteristics, right?
Yeah, he's doing all the positional isomers of mescaline.
Exactly.
The 3,4,5, 2,4,5, 2,3,4, basically mapping that out.
Yeah, so this is the first thing that he did. So, and then what else did he compare? I think that there is a table somewhere here which I will try to find. No, it was not in this paper, or was there? I think there was at least one table. No, it was in the second paper from 69. Yeah, this is the table that he published in the other paper from 69, structure activity of wandering psychotomimetics, and he says that these are all the new as well as previously known psychotomimetic compounds of the phenethylamine type. Yeah, he lists them without giving names. So, but there is like about 40 something, 44 of them listed, and they are compared mostly with relation to the activity of mescaline, yeah, in mescaline units. This is the most important criterion for him, yeah. Activity in comparison with mescaline. So this is not really a qualitative, it's a quantitative relation, yes. How many times more potent certain compound is, right? And I think this table is a similar one, was wait, also repeated in subsequent papers, which I will try to show now, perhaps. Where is it? I think it's in the handout. Yeah, maybe I can share it with you. Oh, I have included it here. All right. So it's also in the presentation. So the same list of substances is given in 81 in the paper hallucinogens from Berger's medicinal chemistry, and you can see the same table as in the 69 paper, right? With names listed. So he starts with TMA, then DMA, then various MDA and MDMA types, then the Aleph family, Paradot, Aleph 1, and so on, and then DOM, DOET, and so on. So, for some reason, the names were not listed in the 69 paper. I have no idea why. Then he lists them. And this is a similar table is repeated 3 years later in a article by Nichols, where he also lists them, and he also has a paper on structure activity relationship. But what I mean is that the only comparison here is done in terms of potency relative to mescaline, right? And other factors than potency are not included. So it's not really a qualitative analysis, but a quantitative one, right?
I think it also might be related to the fact that journals back then, and perhaps even more so today, are unwilling to publish in thorough detail information on the activity of psychoactive drugs.
Okay.
Especially concerning to the content of the experience.
Oh yeah.
And so this might have been perceived as scientifically narrow enough to be included. Although I'm not sure what's the situation at this particular time, either the 69 or 81 or others. But Sasha did mention that towards the end of his life, journals started to reject his submissions for publications on the activity of human data of drugs. And it certainly is the case today. So there might have been a role because it simply wasn't possible or
Yes.
hard. Or also it might have been due to the fact that, as we've discussed in previous seminars, also Sasha in the beginning was interested mostly in the potencies.
Yes.
Since he thought that was, the way to compare these substances that he creates. And throughout his life, further changed to consider the content of the experience and the interactions of the experience and humans' lives and interactions between humans as much more significant and valuable.
Yeah, and the phenomenological data, the qualitative aspect was described in Tikal and Pikal, which is what's missing here, because in fact you cannot easily compare qualitative data. What you can compare is quantitative data. That's why it's scientific, as you said, because you can measure it and compare it, right? You cannot measure qualitative data unless you have some kind of scale, which, of course, we have this Shulgin scale, but in fact, it's like with wine. You cannot really compare the subjective effect that someone, the subjective uh, 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 feeling that you have, the subjective experience. What you can compare is objective facts, and potency, objective potency can be measured and therefore compared and therefore scientifically analyzed and published, right? So that would yeah.
I would just be careful about these, or let's review how we use the word scientific, because
Okay.
it could be seen as the word coming from the Latin scientia and as meaning knowledge.
Mm-hmm.
And in that sense, I believe the subjective responses, albeit unmeasurable in any objective sense, are valuable knowledge, information, and data
Yes.
that can be acquired in a specific way that is subjective experience.
Agreed.
And shared about through reports and so on.
Yeah, agreed.
But
It's
I
like
consider that very much a scientific domain, just as well as any observation from an outside observer, or things like a potency of the active threshold, which, by the way, also doesn't have to be the same all the time, and different people sometimes have different responses to the same amount of a drug, and it also changes with age and so on. So even this so-called objective information is not invariable in any sense.
this is true, and the argument about the etymological argument about the term science, I think it's Sasha's own argument we've mentioned it here before, but it's also true for the Greeks, from the Greek perspective, ancient Greek perspective on science. I mean, the Aristotelian one, where science is something as episteme is something that also includes subjective experience of something. Yes, it does not have to be only measured objectively with scientific method by means of induction and generalization of various measurements, right? So yeah, I agree. Science as attainment of knowledge, also by means of experience, is completely mm, mm, uh, in agreement with the ancient Greek concept of science. So you're right, but that could perhaps be not, for example, Sasha's own standpoint, but the standpoint of the journals that publish, as you said, the articles. Perhaps.
Yeah, I think one thing that's important to remember is that Sasha stated that he was basically only after one question. He was only interested in creating new psychedelics. And many times when he makes a change somewhere on the structure and it has a downturn in either what he feels is psychedelicness or general potency,
Yeah.
he'll abandon that position and start working somewhere else. A good example is the alpha substituted mescalines. Uh, the first is is a TMA, and then so he gets the idea, oh, let's put an ethyl chain there, and very quickly he realizes like the potency's dropped off, and even though he's made and named the next 8 or so chain lengths at that position, he just pretty much abandons the whole project and never gets around to tasting any of them, never remakes any of them. I forget where he moves from then, but yeah, so, and then also just the Shulgin scale is only qualitative. It's hard to understand the Shulgin scale if you don't understand that Sasha's only after, is it psychedelic? And if it is, what's the potency related to compounds we already have? And so the Shulgin scale is basically just a key to whether you feel the full effects of the drug have been reached.
Yeah.
Right, which is the 3, and 4 is just a category because once it happens a couple times, you should probably have a category for it. But 2, 3, 4, and basically just 3 is yes the full effects of whatever this compound does. It's just what he was looking for it to do was be psychedelic.
Yeah. Yeah.
And so I'm not sure if you could reach a 3. It's very hard. It gets a little goofy at the upper levels, but he definitely didn't do enough talking about that. But it's important to remember that. I mean, Pical is full of what look like losers compared. If you look at him through this metric that Sasha is, which is it psychedelic? I mean, most of the compounds he's not that interested in. But a lot of them, if you go look at them, they sound really interesting. They just never reach full psychedelia. The perfect example is Ariadne. It's not psychedelic. It doesn't really get you high in that way. And I somewhere I think maybe in the Toms or near Tomso, Sasha even states in Picel that he knows there's something about these other non-psychedelic drugs. There's something interesting. He can't see it. He's aware he can't see it. But he just kind of never runs out of new psychedelics to find.
Yes.
And he pretty much develops the 2-4-5 substitution pattern. I mean, that's pretty much doesn't exist in nature like that. There's no, the mescaline are 3-4-5, and so he kind of just once he discovers the 4 position of the 2-4-5 substitution, he kind of just never runs out. But yeah, it's important to remember. I think that he's only looking for psychedelics, and that's what he's just abandons any changes that seem like they're going in the other direction.
Yeah, I think we agree in this point. So how about having a look at the principles of structure activity relationship that he introduced in those papers, in the first papers that we are looking at. So I think it's in the first paper, psychotomimetic agents related to catecholamines. It's page 17, 18, 19, I think. And to look at that generally, what does he say? Well, I think in the paper, when I look at it right now, he says that certain structural features appear meaningful in the interpretation of the activity values, which can be assigned to them. It's on page 17. One of the most apparent of these parameters is the length of the side chain. Yeah. And so he says, why is the side chain length important? He says that although mescaline is the primary amine, it appears to be completely ineffective as a substrate for the monoamine oxidase enzyme system, yeah, that metabolizes the compounds and makes them inactive, which is responsible for the oxidative deamination of most phenylethylamines. This inertness may be due to the presence, so here's a structural hypothesis, due to the presence of an extremely basic aromatic ring, which serves as a second base site and renders the molecule immune to metabolism. And so, for example, various analogs of mescaline, like DMPEA, yeah, that he examines here in those papers in detail because of its presence in the urine of schizophrenics, right? He says various analogs of mescaline are readily deaminated by the enzyme systems and therefore shown to be inactive in humans. So the question for him is how to circumvent the enzymic destruction, yeah, and therefore making the compound active. For example, by adding a branched methyl group on the carbon adjacent to the amine group. So by lengthening the side chain, yeah. So what he tries to do is either to make a compound more potent or active at all, right? By somehow circumventing the enzymatic system, yeah, the monoamine oxidase system. So this is one thing. And then he mentions the second thing are those various positions, the positional isomers, and his first experiment with modifications in the TMA family of compounds that results in the conclusion that the 2,4,5 orientation is the most potent of all, right? The 2,4,5,3-methoxy or TMA2 is the most potent, and in particular more potent than 3,4,5, that is masculine, right? So the 2,4,5 orientation is most potent, about an order of magnitude more effective than the 3,4,5 isomer, that is masculine, and it still retains much of the qualitative nature of masculine. Yeah. So these are two things that he notices in this star structure activity relationship analysis. What else? Another anything to comment on it or not? Because then on page 18, he mentions another possible substitution, methylene deoxy substitution, yeah? And here there is a qualitative aspect. And he says, then in this case of replacing of two adjacent methoxyl groups with a 5-membered heterocyclic ring that we know, for example, from the structure of MDMA, right? If you take two methoxy groups adjacent to each other, and if you connect them by forming a second ring, yes, then a strange thing occurs. And of course, he did it because of his work with nutmeg and other natural substances that have this ring. So he was kind of, you can say, combining various structures found in nature. So he took amphetamine and put it on top of mescaline, which is one thing. Another thing was taking structure from nutmeg and other similar substances, which we've discussed, and which he described in the 60s and examined in the 60s, of course, right? And create another family of compounds with this 5-membered heterocyclic ring. Yeah. So this methylene deoxy substitution introduces, as he says, very interesting qualitative features, not only quantitative, not only in terms of potency, but in terms of subjective qualities that he says that are not really similar to tryptamines, to the profound religious insights so characteristic of the indolic psychotomimetics. Yeah, so they are completely different. There is a a loss of the property generally accepted as psychedelic, but in its place there is a form of a dream world. A dream world. Yeah. Either dream-like entertainment or a dream world structured in the framework of the past. Okay, but then those methylene deoxy chemicals with this second ring attached are of course MDA, MDMA, and other substances, and he compares them in terms of potency and in terms of effect, and notices already here in 1969 that this property of apparently valid memory recall, coupled with the capability of maintaining normal behavior, can be useful in psychotherapy, of course. So here already he realizes this therapeutic potential of this group of phenethylamines. Okay. Any comments on it?
Yeah, I think to summarize, he's just kind of basically laying out the three generalized effects that you can get with the phenethylamine. You can get the psychedelic type effects, characterized by like DOM stuff, 2Cs. You can get the amphetamine type effect with the alpha substitution, being the main character there. And then you can get this third kind of pathogenic and tectogenic effect, usually with this methylene doxy bridge. That'd be your MDMA, MMDAs, all those. These are kind of your three poles of the effects you can get with this basic structure.
Yes, and there's the another possibility mentioned, namely replacement of the alkyl group, yeah, which increases potency by replacing the alkoxy group with alkyl group, which results with the OM, the OET, and so on. And so this is called here steric blocking. Maybe you could explain why it's called that way. Why it gives metabolic stability. Why, how and why it increases potency. This is on page 18 in the paper. He says that the rationale that dictated such a substitution, substitution of the alkoxy group with an alkyl group, is the rationale, the reason, the justification is simple. Namely, in the few human metabolic studies conducted with these materials, it seemed consistent that detoxicative conjugation was preceded by either hydroxylation of the aromatic system or by the methylation of the methoxyl group. To generate such a hydroxyl group, it seemed clear that if a vital position could be blocked, a vital steric position could be blocked by a group that was not amenable to hydrolysis, the biological nature of the compound could be radically altered. The compound that would result might be more potent, and it cannot be readily disposed of through normal metabolic procedures. On the other hand, it could be without effectiveness, but might serve in turn as an antagonist to the action of the other materials chemically related to it. Yeah. So the first example he says confirmed the former possibility, and therefore the methyl analog of TMA2 is the most active of methoxylated amphetamines, and it led to DOM. Yeah. So he has a reason it was not working blindfolded. Yes, he had a very strong reason for doing certain changes, structural changes, and examining their result, right? Yitka, would you comment on it?
I mean, also with the DOM, it was such a breakthrough for him because, as you just read this commentary, what was the hypothesis behind designing this compound was a surprising discovery, which then led to many further directions along this line, and also implying that this proposition later on seemed to have proven to be this point where the effects are really modulated. So I feel Sasha held this compound really dearly close to him because it just shaped the direction through his research and his understanding of how these compounds work, at least if we consider on a biochemical level how they work.
Yeah. On the biochemical level, he knew that this compound would be inactive because of certain metabolic processes that could be blocked by means of a certain change in the structure, right? So this was done, and this hypothesis seems to have worked and generated a new family of compounds, right? So this is how DOM and DOET and other substances of this family were created.
Yeah, it so shocked him that he set off on the 10 classic ladies, which was a process of replacing each of the 10 unique hydrogens on DOM with a methyl group in an attempt to understand why the potency was so much more. Yeah, I think, because hit on it. This surprised him. You make changes with a rationale, but occasionally you just get surprised. And yeah, the 10 classic ladies, some of which he made, he never got around to tasting, but the most well-known, of course, is Ariadne.
But why do you think they were not really tasted or?
Well, I think what we're seeing here in this paper is kind of Sasha's, and Yitka was getting to, is like Sasha's maybe first written words where he's starting to zone in on this magic, what he ends up calling the magic 4 position, that leaving intact the 2,4,5 substitution pattern, leaving in fact the 2 and the 5 methoxy groups, and just changing the 4 position. And then also with alpha substitution, so all the amphetamine analogs, sometimes before the 2C analogs, sometimes after. And that seems to have occupied most of his time. Was just kind of like realizing, the 4 positions where the magic is. There's a little bit of unsymmetric work. Some of the classic ladies are unsymmetric. Iris. But yeah, he kind of just never really exhausts. I mean, once he starts putting sulfurs in, he kind of just never really exhausts what he could put at that 4 position. And realizing he kind of maintains the psychedelicness, and then is just kind of changing the flavor a little bit with the 4 position. Something he later applied to kind of the tryptamines. The last compounds he was working on were these DALTs, these diallyl tryptamines. And he never really got to finish that project, but he made some predictions like, I think if you left one of these allele groups and just changed the other, it would be like changing the 4 position. You kind of keep this short duration tryptamine, and you could just kind of vary the flavor. And Nasser, did a bunch of that work, and I mean, he was pretty much right on all of it. So kind of interesting.
Sulfur, you mean the Aleph family?
The Alefs, yeah, but eventually the 2CTs, basically, which are the phenethylamine counterparts of the Alefs, which didn't seem to have the wide dosing range problems of the Alefs, which is...
Yeah.
kind of unsettling to Sasha, but he found the two C's didn't really, kind of lack that problem, and were maybe more special and psychedelic.
Yeah. All right. So perhaps we could proceed to the second paper and then have a look at the progression, uh, at the differences between those papers that we are examining. So in the first paper, this is what he discussed, and also he has those claims on DMPA, right? There is this hypothesis of endogenous substance that could have resulted in some kind of psychedelic or psychotic activity. Yes.
Yeah, it was kind of like a popular idea at the time. I think it fairly quickly gets abandoned.
Yeah, but still he considers it here and in the series of subsequent papers. Even in I think one of the most interesting tables that if you maybe we could look at it right now, and let's just skip the second paper from 69. Maybe we can return to it in a while. There is this very interesting table which I will display right now. And it could be interesting to have a look at it and try to understand it. It's let me tell you, it's from the 1970 paper, chemistry and structure activity of the psychotomimetics. It's on page 31, and there is a comment on it. Here, the comment is given in the paper on page, let me tell you, 36. So what are there? All those compounds that he analyzed are listed here. And this table, it is a very strange one because he suggests that there is a relation, an internal relation, metabolic relation between all those compounds. Yes, I'm not sure if you have looked at it, but to me it is very interesting. Perhaps you could try to interpret it somehow. So in this paper, he makes much more. Let's look at it from the beginning. Much more general claims on the structure activity relationship, because in the other one from 69, structure activity. Sorry, there are two more papers. Maybe let's look at them first. The structure activity of wandering psychotomimetics. It gives the same examples. It also has this hypothesis of DMPEA and internal metabolism, and he gives the same examples. The same relations are described as in the other paper from 69. So he has this side chain length factor. Yeah, the longer it is, the more potent it is. He mentions alpha methylation. Yeah, of course, methoxy position is crucial, and the 2,4,5 position and 3,4,5 position, and the methylene deoxybridge, as in MDA and in MDMA, also not only increases potency but changes the quality, as we've mentioned. Yes, and the same with the DOM family, and also and one more argument given here is the possibility of indole formation. Yeah, that he says that there is a possibility that indoles could be formed out of phenethylamines by means of creation of another ring, yes, in vivo, so by converting from catecholamines or phenethylamines, yes, with a certain structure of the benzene ring to indoles, which are then active psychedelically, right? And this is developed later in the third paper. So this is interesting, I think. So this indole possibility is already stated here in the second paper. All right, and then it is developed further on, and I think we could discuss the details, but I don't think we have enough time to do that. So the only thing that maybe I would like to discuss at the moment is this third paper, Chemistry and Structure Activity, the 1970 paper. And so perhaps not not to discuss it in all possible details because this is too far-reaching, but this table where he mentions and describes the possibility of indole formation. This is, I think, the most interesting thing of this paper. How about having a look at it? What do you think? I can read this passage. Maybe that would be helpful. Let me try to find it. This is the passage. Look. That suggestions have been advanced that these materials may show biological activity, yes, due to the fact that they may imitate or form indoles during the course of their metabolism. And what materials is he mentioning? Of course, he means phenethylamines here, various phenethylamines, those materials listed here on the left, masculine TMA, MDA, MDMA, and so on, COM. So he suggests that they are converted to indoles and therefore they are active ultimately, yes? So there is a hypothesis about the internal route, about an internal conversion that takes place, and he shows in this table how they hypothetically could be converted ultimately to an indole compound.
How could you test this hypothesis? Put some in a mouse, then kill it and freeze dry it, and look at the brain.
Yes, is this Steve? Or who is it talking? Yes. I think Steve started making a comment but didn't manage to.
You know, I don't know much about biochemistry or chemistry, but
Yes.
my understanding of this was it was like an attempt at the time to explain how such structurally different compounds were eliciting the same effects. How was something like mescaline and TMA2 LSD like when the structure indicated a more amphetamine kind of effect? And
yeah.
I think what happened is that just our understanding of brain chemistry got more sophisticated over time.
Yes.
And this theory just kind of isn't necessary anymore. That's kind of my understanding of it.
All right. Okay.
But the table is certainly very interesting. There's obviously a lot of information packed in here. I don't know that anybody here could do that on the fly. But yeah, I think he's mapping out the possibilities for this.
Well, clearly it shows a structural relation between these compounds, how they are converted to one another, right? But then he concludes that in the last sentence, on this basis, one can conclude that quinone formation is not a likely explanation, right? So does this mean that this hypothesis is falsified and rejected? I'm not sure. It's interesting. I would like to hear a comment on it, but maybe not necessarily at the moment. But certainly this is a progress from the previous papers, where such a hypothesis was only suggested first in the structure-activity relation of one-ring psychotomimetic, and here it was developed. All right. So this is something that was not suggested before, and theoretically researched. And that, yeah. Anything else on this third paper? It is chemistry and structure activity of the psychotomimetics. It is a huge, way longer than the previous papers, and it contains much more material, chemical material, and the comparisons are more extensive. All right. So he mentioned, well, all those substances that were examined in the 60s, and he creates a theory where he not only discusses the differences in potency, right? So in the paper, I think what interests us is on page 30, he starts with, or even on page 29, he has a brief description of tryptamines. He starts with DMT, then 5-MeO-DMT. He already here he describes DET and DPT, diethyl dipropyltryptamine. He mentions also 5-HO and 4-HO DMT, and this is it. So there is just a brief discussion of what he will then expand on in Tikal. But then the most interesting part of this article is this general description of phenethylamines. So first mescaline, of course, then he describes in a very interesting paragraph how and briefly and extensively how he experimented with TMA, yeah. Then with after examining nutmeg, he started doing the methyl and deoxy ring, adding the second ring to the basic phenethylamine structure, and this is what was already discussed here. And then he goes to tetra MA to DMA. These are other structures that he experimented with. Yeah. So this is the meta, ortho, and para rubric in the table that we have seen here. Yeah. second and the third column on the right. Yeah. So this is something that was not present in the previous papers. And this para and ortho substitution is something, is a novelty in this paper, which I think could be discussed. And there is a final conclusion that I can perhaps read at the moment. He says on page 37 of the paper, what can one conclude from all this? The ultimate purpose of this study is to look at what compounds might be present in the human, not through normal metabolism, but generated through some idiosyncratic or pathologic state. This material might result from some norepinephrine pathway deviation and might be a biologically active agent, although it appears that a chain length of 3 carbons is optimum. This may merely reflect the defense of the amino group to deamination. As 3 carbon chains are virtually unknown in animal metabolism, it is probable that the 2 carbon chain in metabolism is a more likely structure for such an agent. An increase in the number of the methoxy groups leads to an increase in the potency of a compound, but this must be carefully analyzed. The addition of ortho groups increases activity. The addition of meta-methoxy groups decreases it. And although the para substituent is not essential, it certainly is of much importance. Yeah, and then he goes on. Yeah, there is a paragraph which I will skip. And a final conclusion, how can one challenge these proposals? Paradoxically, the most direct way of challenging this entire group will fail. For if you have a material that is suspected of being an endogenous psychotogen, you cannot assay it in normal human subjects because the normal subject is indeed normal, for the very reason that he has the machinery for disposing of such a compound. Therefore, one must distort such a compound in some way that will make it a valid challenge. One way would be through the introduction of a methyl group, group alpha, to the amine function, which is what we mentioned. This would lead to a series of variously substituted ephedrines, presumably less susceptible to a monoamine oxidase attack. Possibly the replacement of the methoxy group in the para position with a methyl group would interfere with metabolic disposition, which we would also like to achieve. And finally, a chemical explanation of mental illness is only one of several that are possible. But the fact remains that there are some aspects of mental illness that can be duplicated chemically. Yeah. So then he concludes that a hypothesis of an endogenous psychotogen certainly deserves further investigation. Okay. So anything more on this paper, or shall we proceed to the last two that we would like to discuss today? We still have a few more minutes because there are 2 more papers. One is the largest one. The 1978 paper, Psychotomimetic Drug Structure Activity Relationships. It's a huge paper from a handbook of psychopharmacology, and it contains a lot of material, and of course we are not going to discuss it all, but the question that I would like to ask is what novel materials does it contain? It's certain, and the last paper from 94, it's extraordinary, I would say. Not only because of the amount of material, empirical material that it generalizes, but maybe we can discuss it in a moment. So how about the 78 paper? It is interesting because it is a systematic presentation, right? Systematic presentation of all important and known at the time, not only phenethylamines, but also other families of compounds. right? So it has not only phenethylamines, but also tryptamines and other compounds, but it mostly focuses on phenethylamines and describes them in such detail as he never before attempted to describe them, including the structure-activity relationships and all various subfamilies, right? So first there is mescaline and natural sources of it. Then there is the second group, phenethylamine. The basic structure, namely mescaline, is on page 259. And then there is the TMPEA, various kinds of 3-methoxyphenethylamine. Then there is 4-methoxyphenethylamine, which is MPEA. Then there is this DMPE, dimethoxyphenethylamine, related to the various forms of schizophrenia. Yeah. And then after discussing mescaline and TMP, DMP, and so on, he proceeds to proscaline, to escaline, so other homologs of mescaline, which were never described by him before. So this is the first paper of Shulgin that describes those homologs of mescaline, like proscaline and escaline, right? According to my knowledge. And then he goes to methylene deoxy compounds like MDA, and so on. And page 272 and the following pages are interesting, because here he gives a general theoretic glance that he calls structure activity generalizations. Yes, and I don't think we have time to discuss it in detail. But then he describes various nitrogen substituted phenethylamines, and then he proceeds to phenylisopropylamines, to methoxylated phenylisopropylamines, that is PMA, DMA, TMA, all those compounds. So there is a structural progression: TMA2, TMA3, TMA4, TMA5. So what is interesting is that they are not listed in an alphabetical order like in PCAL, but they are, so to speak, developed structurally in a progressive order. Out of the basic structure, there is a structural progression. Then he goes to methylene deoxyphenylisopropylamines, and that includes MDA. First, of course, he starts with safrole and myristicin and other natural compounds that have this bridge, this second ring, and then he derives MDA, MDM, MDE, MMDA, MEDA, MMDA2, and so on, others, DMMDA. So all of them are derived and described in detail in a systematic order. And then he proceeds to alkoxyphenylisopropylamines, that is MEM, EMM, MME, MPM. When you read the lab book, they are there. He experimented a lot with them, and those experiments with this family of alkoxyphenylisophenethylamines are not described anywhere else in any other paper of Shulgin until this one. They were mentioned in the lab books, of course. So then we have alkyl phenylisopropylamines, and they include DOM and other derivative compounds. There is a huge chapter on DOM. Then there are other compounds from this family like DOET, DOPR, and so on and so on. DOBU, DOAM, and then various halo and sulfur substituted compounds like for CA and others, then BOB. DOI, I will not read them all at the moment. And then there is Paradot and the LF family. So all those compounds that he ends with Paradot and LF. All right. So all those compounds that are listed in an alphabetic order in Pical are here given described in a structural progression where he begins with the simplest compound, and out of it he develops all of those secondary subsidiary families. They are this. I think this is the crucial thing, derived structurally and systematically, which never before has been attempted in this way, and also never again in those large books of Shulgin like Tikal and Pikal. So it's an amazing paper from this perspective. What do you think?
Yeah, I agree. The one thing about Pical's organizational structure is it has none. It's an alphabetical. So
yeah, exactly. That's what I mean.
This is a welcome addition for sure.
For the sake for understanding for to understand the structure and the relations, this is crucial. This is crucial, I think, to organize.
Yeah, I agree. You get kind of the more complete understanding of Sasha's grokking of everything here.
Yeah,
it's in Pical. It's just all spread out and not all in one spot.
Well, there is this famous review that is circulating in the internet. This review of Pical, which was I think published in various websites. It was called A Structural Tour of Pical by R. Kundalini. You certainly know it. And what it attempts to do is to reconstruct this structural progression on the basis of Pical. But apparently the author of this paper, R. Kundalini, who published this structural tour in the year 2000, he, so to speak, derived the structural relation out of the content of Tikal itself, not knowing that Shulgin himself did it about 22 years before. So he repeats. So of course it is kind of implied and assumed by Tikal, and somehow possible to reconstruct out of it, which is what this person R. Kundalini did, attempted to do, but he never even managed to match the detail and the depth of analysis that Shulgin himself achieved in this 78 paper.
Yeah, I believe you can get that through Isomer Design's website. There's a lot of good stuff there.
Of course.
A lot of us end up doing this. I still have a version of this I'm doing myself called the Technical Edition, the PTED. It's kind of like Terence McKenna said, the only understanding of the universe that really matters is your own. So putting together a document like this can do a great deal for one as far as kind of internalizing everything Sasha learned in a way that you can then act on.
Yeah, it's a fair comment. I think I agree with that. Yeah, as in this proverb that there are only two kinds of people. There are those who are self-learners or who are no learners at all. In Polish, it's like so ludzie dzielą się na nieuków i na samouków. That you divide people in two categories: those who don't learn at all, or those who learn everything on their own, right? So you have to do it on your own. Agreed.
As I'm sure you've learned, putting this amazing group of documents together.
That's what we all do.
Yeah.
So the last paper, and any comments on it? It's written with Peyton Jacob the third, and I don't know to what extent it was written by Sasha himself. It seems that he's listed as the second author, but all those materials, information contained here shows that it was written after not only Pikal but also after Tikal. There are so many it was written apparently 3 years before the publication of Tikal, but all those tryptamines described in Tikal are already included in the analysis done here in this paper. So this generalizes the knowledge that we have upon the basis of a way richer and wider empirical base than the previous articles. Yeah, so it seems like this is the ultimate statement of Sasha on the subject of structural activity relationships, and it has so many insightful claims that I don't think we can even attempt to discuss them in detail, but it shows how what knowledge he achieved. But so maybe the only question is, does it even make any sense? Is there a point, I would ask, of theoretically analyzing? This is the question to you, maybe the final question that we can answer. Is there a point in theoretically analyzing the different structure activity relations without attempting to repeat the synthetic path, without synthesizing those compounds and looking at the result on your own? I mean, this is there any sense because Sasha himself concluded this as a result of his own practical experiments, practical research. So to rephrase this question, is there a point in studying this theory of structure-activity relationships unless this theory is rooted in practice, in chemical practice? So, in other words, perhaps this theory makes sense only if you do generalizations out of practical chemical work. Otherwise, this theory is detached from chemical practice and has no meaning, or at least no usage at all. What do you think?
Yeah, I mean, I think you could argue that there's limited usage. Humans love this, though. Baseball stats, and I mean, this is not uncommon to be interested in a bunch of stuff. I think it certainly gets much more interesting when you start doing the bench work. At least as far as I mean, nowadays it's a lot of applied research, right? So you're kind of like looking for something in particular, even if you have a general idea. As opposed to Sasha. I mean, I guess you could say the same for Sasha, but I mean, as far as following up, Sasha left an awful lot of breadcrumbs and an awful lot of go this way signs.
Yes.
He certainly didn't have enough. So I think once you're doing the bench work, to be studying what he found, is going to help. But it's also important to remember that SARS are like a working thing. It's not, you're never gonna, it is the rule until it's not. You know, MDMA is
Right.
one of these drugs that breaks all the rules. The N substitution should make it weaker. The S isomer should be not as good. I mean, with all the other phenethylamines, it seems to be the R isomer where the action's at. Here you have a compound that breaks all the rules and is something really special. So SARS are great to kind of guide you, but in the end, until the compound is made and tasted, you don't know if the SARS holds up in that with that new compound.
It is a kind of a map, as you said, and a way of, let's say, uh, raising the experience obtained in practical chemical work to a higher theoretical level, where you look at this entire field, all those paths that you have uh, gone through, and you look at them and you kind of draw a complete theoretical map. Yes, not only describe a certain compound, how you synthesized it, what characteristics it has, but so, to me, SAR is a kind of um, a report and a map and a general attempt to view the entire field that you have gone through, yeah, that you have explored, not only to give a report, but also, as you said, to give an advice or a guide how to continue this route of chemical uh, research of the human mind ultimately. Yes.
Yeah, exactly. It's a map of the coastline that's been seen so far,
Yeah.
and it helps you give an indication of where you might find certain features that you're looking for. um, you'll get there quicker doing this than having an AI generate you 30,000 ligand leads. I mean, to me, that's ridiculous. Like, how does that help? Now you've got a list of 30,000 things that need to be made and tasted. I mean,
Yeah.
you'd be better off, I think, and quicker to the target if you
Yeah.
study just Shulgin's kind of thing, use it as a guide to where you might find the features you're looking for.
Agreed. Unless you feed Shulgin's SAR papers to AI, then it could maybe make some use of it and not only do random statistical generation of all possible combinations, right?
Yeah, but the problem is that in order, this compound still has to be made and tasted,
Yeah.
because the AI can only know what we know, and we have a very rudimentary understanding of human brain chemistry. So, until until such time that's complete, the AI is going to struggle with all the same things we're going to struggle with. I mean, I think a human could probably predict all those same things, but at some point, there's like a gut. You got to kind of go with your gut towards the things, and you got to make a few compounds and see if you're kind of getting results you want.
Yeah.
Kind of feel your way through the dark eventually, anyway.
Agreed. Agreed. Yitka, any final comments? Yitka, are you there with us?
Yes, my phone is slow.
So any final
Mark,
comments on your side?
it's good as a guiding work to get your ideas based on whatever you wish to be creating, unless it's completely open search of curiosity, see what is here, and let it be shown to me as I make it. These reviews are of importance for catalyzing or facilitating some kind of a design of where to go next based on what you want or where your heart is.
Yeah, I think, for example, Traxel's book on phenethylamines, not translated into English yet, is an attempt to go perhaps further and certainly to do what Sasha did here in those structure activity relationship papers in a more complete way, right? Other than that, there has been no attempt, according
to bring it up to date. It's been a long time since Piccal was published. A lot of chemistry has been done.
Exactly. But no one did that with tryptamines. Only phenethylamines have been described in a complete synthetic way in by Traxel. But there is no structural analysis of this kind of tryptamines so far, as I know. Would you can you confirm that?
I think you're right. These are a lot of these are also a lot more recent work with tryptamines and isotryptamines, and you're more restricted on your ring substitution, basically to 5 and 4. But there's other substitutions. The isotryptamines are really interesting, where the chain comes off the 5-membered rings nitrogen instead of off the end of the chain. 2 position. There's been some work there, but yeah, that'd be a volume worth having.
Somebody will produce it sooner or later. Yeah. Okay. So thanks for this discussion, and next time we discuss Piccal and Tikal, which is going to be very interesting, I think.
Thanks, guys.
Thank you.
Thank you very much. See you next time.